Welcome back, everyone, to this final recording on the adaptive immune response and on the whole topic in general. Here we're going to learn about the actions of antibodies. So we need you to be able to identify those, be able to describe how vaccination works, and compare the ways that immunity is acquired. So just a little bit of a recap on what I was talking about in the other recording. What we want to learn about here is how we end up with all of these antibodies over here. So let's go back here. We've got a bacterium being engulfed by a macrophage, maybe being that antigen presenting cell, it being broken down, and then those antigens then combining with an MHC class 2 molecule being presented on the surface of the cell. We've got our T cell receptor recognizing the antigen, and our CD4 molecule of this helper T cell binding to the MHC class 2 molecule. The net effect of all of that, right, is this activated T cell. At the same time, we have B cells, okay? Now, the antigenic receptor for the B cells, remember I spoke earlier, is just an antibody in the plasma membrane. So these B cells are going to bind the pathogen too, you know, if they have that receptor for the antigen there. And then these activated T cells will then bind to the B cells, and through the process of releasing cytokines, that will then activate this B cell, cause it to, you know, proliferate and differentiate into this clone of plasma cells that then produce these antibodies. And these antibodies, right, are the same as this antibody that bound the bacterium in this case to begin with. And of course, we also have this clone of memory B cells being formed, and they're going to be really important then in that secondary kind of antibody response if the person gets exposed to that same bacterium at a later date. Now, remember that we have this primary response and secondary response that I spoke about earlier. So here we've got antibodies on the y-axis, time on the x-axis. And here we have an initial exposure to an antigen, right? And here we have this primary antibody-mediated adaptive response, which you can see takes time, and it's the magnitude of it is quite low as well. All right, so the primary response occurs when a B cell is first activated by an antigen. The B cell proliferates to produce plasma cells, antibody production, and memory cells, but it is low and it's slow. Okay, and it's during that time, you know, if we have been infected with the pathogen, that we develop the symptoms of the disease. On secondary exposure then to that same antigen, you get the secondary immune response, right? And because we have these memory cells, they can divide really rapidly to form plasma cells and additional memory cells. So you get a much faster response and a much greater magnitude of response to neutralize and eventually destroy the pathogen. So antibody-mediated immunity is effective against extracellular antigens, so those outside of cells. And this includes bacteria and viruses and other types of microorganisms, and also toxins. Antibodies are also known as immunoglobulins, and they're abbreviated to IG. And there are 5 classes of them: IgG, IgM, IgA, IgE, not to be confused with the supermarket, and IgD. You can see their structure down here. They have a variable region, and that variable region is indicated there. They've got an antigen binding site there. So that, you know, the variable region is the part that combines with the antigenic determinant of the particular antigen. And then we have this constant region down here, which is responsible for activities of antibodies like activating the complement system or attaching to various kinds of white blood cells. And so here, this is just the usual schematic that you look at in a textbook, and this is a 3D representation of IgG. All right, so this is just a table showing the 5 immunoglobulin classes or antibody classes. We don't want you to memorize all of this stuff. Just know that there's 5. Know that the IgM is very large. It's very obvious there. And it's the main antibody of the primary response, right? It's the best at fixing complement. Okay, and that's all you kind of need to know. The IgG then is the main blood antibody of the secondary responses, and it neutralizes toxins, and it also performs that opsonization, where it binds to the antigen, and that makes it more visible for phagocytosis. And then, this one's an interesting one, IgE, because it's the antibody of allergy and antiparasitic activity. So people who have allergies, this antibody, when it's produced, it kind of coats itself on mast cells. And then whenever the allergen, or whatever they're allergic to, say it's pollen, comes along again, it binds to these IgE antibodies on the surface of mast cells and causes them to wriggle around, and that releases all those granules, and those granules contain things like histamine that causes the allergic reaction and the wherever it is in the respiratory tract to cause bronchospasm. So to summarize the actions of antibodies, you can see these pretty pictures here. So antibodies bind to antigens, and that inactivates antigens in a couple of different ways, as you can see here. Neutralization is the first one. So here we've got these antibodies binding to viruses and bacteria, and that blocks the viral sort of binding sites and also coats the bacterium. So that neutralizes the antigen. Can also cause things like agglutination of microbes. Okay, so here you can see all of these antibodies that sort of link up together and bind these bacteria, in this case, to cause agglutination. And remember, we used that agglutination process to work out our blood group, didn't we? Precipitation of dissolved antigens as well. So you can see them all sort of, those antibodies sort of linking them all together to cause precipitation. Also activation of the complement system. So, and that, once complement is activated, and you get that series of subsequent activation of the different proteins of the complement system, that then causes, that punching of a hole through the plasma membrane of a particular pathogen. Now, all of these first three enhance phagocytosis via a macrophage. And that enhancement all falls under that sort of category of opsonization. So then you can see it's much easier for the macrophage then, to engulf or phagocytose this antigen-antibody complex. And as I mentioned, the activation of the complement system leads to that cellular lysis. The final thing we're going to look at now is how we acquire immunity. And immunization itself is the deliberate introduction into the body of an antigen or an antibody. Two different ways we acquire immunity broadly. It's either active immunity or passive immunity. And there are two categories of active immunity and two for passive. Okay, so the first one is active natural immunity. Now, with active immunity, it's your immune system that's doing all the work, right? So active natural immunity is natural exposure to an antigen, whether that be, COVID or measles or chickenpox or whatever it is, right? If you are naturally exposed to that antigen and you haven't been vaccinated, then you're obviously going to get sick, right? And it's during that primary immune response then that your immune system develops immunity. And obviously that's the one that's slow and it's low, but it does result in the production of those memory cells, so that then when you get exposed to those antigens again, those memory cells rapidly divide and clears the pathogen before you may not even know that you've been exposed to it. Active artificial immunity then is via vaccination. And with vaccination, and I'll talk about this more in the next few slides, there is deliberate exposure to an antigen. And that antigen is the vaccine. And the antigen is a part of the microbe or whatever it is that you want to stimulate your immune system with. So you don't get sick, you don't get the symptoms of the particular disease, but it's enough to stimulate an immune response and you develop antibodies and all of that. So this is active artificial immunity. An active immunity can last, for several weeks with the common cold, or it can be right out, to years and even a lifetime then, say with polio, particularly the polio vaccine. Now, passive then, doesn't require your immune system to do anything. It's literally just the transfer of antibodies. And this includes passive natural immunity and passive artificial immunity. With passive natural immunity, where we see this is the transfer of antibodies from a mother to her developing fetus, or to the baby, in milk, so in the lactating mother. And that milk then, has those antibodies in it, and that offers the baby protection while their own immune system is developing. Passive artificial immunity then involves the transfer of antibodies or cells from an immune animal to a non-immune one. Okay, so what does this mean? We're talking about anti-serum here, which is antibodies, right? So you would expose an animal to a particular antigen, and their immune system would do the work and produce the antibodies, and those antibodies are then harvested from that animal and packaged up, into a little bottle of anti-serum, and then that's injected into a person when, there's just no time to wait for this active immune response to occur, right? So say if you've got like tetanus or botulism or something like that, where literally, if the primary adaptive immune response takes up to 2 weeks, that's going to be too late, right? In terms of, the person might die while you're waiting on an antibody response. So what you can give to them in the interim is the antibodies, right, to neutralize and destroy the particular pathogen. And these anti-sera are available for things like rabies, hepatitis, measles, toxins formed by bacteria such as tetanus, which I mentioned, diphtheria, botulism, also venoms from poisonous snakes and black widow spiders. The most important thing to note here, everybody, is that your immune system is not doing anything with this passive, natural or artificial immunity, and it's short-lived, okay, compared to the active one. So just to summarize this a little bit more, we have acquired adaptive immunity, can be active immunity, and it can be passive immunity. Okay, if we look down here, we have natural passive immunity and artificial passive immunity. I've just described them, so I won't talk about them again. And they are going to lead to short-term acquired adaptive immunity. And then we have natural active immunity, artificial active immunity, and both of those will lead to more long-lived acquired adaptive immunity. And it just depends on what, the pathogen is, that will sort of determine how long you'll have that acquired adaptive immunity. And for many, diseases, they don't last, the immunity doesn't last a lifetime. So we have to have things like boosters. And just again, this artificial passive immunity is through the use of. So just a little bit more about vaccination now. Of course, vaccination uses a person's own immune system to attack a pathogen in several different ways. Does provide long-term protection, usually years to a lifetime, although I did mention that, the immunity to the common cold is not very long. And I guess that explains why we're always getting colds, right? The substances that we can use in a vaccine include a particular part of a microbe or a dead microbe or a diluted, attenuated microbe, which is still alive, right? But it doesn't cause disease. Now, we've also got these messenger, so they're the traditional vaccinations, right? Or vaccines. Now we've got these ones that are called next generation, although they're being used now. So I think we better drop the next generation. So these are these messenger RNA vaccines. And this is a code of messenger RNA that codes for a viral protein in the case of the COVID-19 vaccine. And I've just popped in that Pfizer and Moderna vaccines, and I'm sure, everybody has had one of those. So what happens here is, you have the vaccine injected into your deltoid. That messenger RNA code gets into cells and it gets translated into the viral protein, okay? And the viral protein then, is transported then to a lymph node where you have that immune reaction occurring. And what then happens is you get a particular antibody and being formed to that particular viral protein, and that's how it works. I think it's always really useful to have a look at how successful vaccination programs have been. So here we're just looking at the number of deaths from currently vaccinated diseases in Australia. It's quite old, but it doesn't really matter, up to 2004. And we're looking at diseases such as diphtheria, whooping cough or pertussis, tetanus, polio, and measles. Now, if you look at the key down here, the red is the last decade before vaccination, right? So if we take it there for whooping cough, so way back in 1926 to 1935, you can see that, 2,808 people died from whooping cough. So per 100,000 of the population, 42.5. Now, look at this in pale blue, which is the commencement decade of vaccination. You can see that really big drop, okay? And then the first full decade of child vaccination, we're getting an even greater drop in the number of deaths. So that's out to, 1955 in that situation, and then gradually dropping off and, pretty much staying stable. Although I will add here that, there has been a little bit of an upsurge in whooping cough, but, that can be sorted by just getting a booster. It seems to be at around about 8 years post vaccination that you can get a recurrence, or you can get whooping cough again. I know because I got it and my son got it, but we got sorted with a booster. So you can follow this through in your own time. You can look at how tetanus has fared with the vaccination program, again, pretty much disappearing. Polio, amazing results with polio, and measles as well. But, probably depending on, I guess, the region of Australia, there are a couple of regions where we are seeing an upsurge in some of these diseases, particularly measles, and that is because of, I guess, the slight increase in people who are trying to, avoid a vaccination. Now, as with every medical intervention, occasionally, thank goodness it is rare, vaccination can cause severe allergic reactions, and the sort of end result of a very severe allergic reaction can be anaphylactic shock, where there's systemic vasodilation of blood vessels, and that can lead to a sudden decrease in blood pressure, and the body can go into anaphylactic shock. Vaccines may contain reagents to help stimulate a strong response. These are called adjuvants, and preservatives as well. And, more often than not, it's actually the allergic reaction is occurring to something in the preservative rather than to the antigen itself. Often people will have flu-like symptoms after vaccination, and that is perfectly natural, and that's a good thing because, it's stimulating your immune response. Okay, everybody, we have finished the topic, so I'm going to say goodbye for now. And when I come back, we're going to have a big change, and we're going to be talking about the respiratory system.